Okay, good afternoon. And first of all, I'd like to thank the organizing committee for inviting me to wonderful symposium and wonderful city of Paris. And this is my second visit here, and I just love the city. Again, thank you. Thank you to chairpersons for a brief introduction. So we have heard about the sirtuins. We have heard about lifestyle changes, lifestyle regulation of the normal aging. But I would like to present some of the data with respect to abnormal lifestyle and also the premature aging. And I take an example of lung because environmental factors are directly related to lung responses, and also particularly in lungs we don't have any remedies. We have a bronchodilators, we have steroids, but they are very temporary, halt the inflammation and as well as the aging of the lung. So first aspect is the environmental stress, how it alters the SIRT1. We have heard the SIRT1, sirtuin-1, and I'll have some introduction of sirtuin in my presentation, and then I talk about the lung inflammation and how it is related to premature lung aging. So as I come out from the metro station, I can see here people have, especially ladies, young ladies, they have smoking, and not only that, actually I come from the background where come from India, where we have a biomass fuel burning. So smoke is the important element which is related to direct, and lung is a direct organ where we can be exposed to environmental insults. So when we have a smoke, and first thing what we see is oxidative stress. It bleaches the epithelial lining fluid, the glutathione, thiol levels, depletes antioxidant. Eventually in chronic smokers we can get upregulation of several phase 2 genes, but that doesn't last longer. And then second step, it comes redox signaling, which is inflammation. And inflammation triggers several aspects of events, for example, apoptosis, autophagy, and cellular senescence. So these are the important things which can lead to premature aging. But some certain cases, we do not have inflammation, but still we get the final outcome, which is cell death or the senescence. So here, so this is the lung, which looks like the normal lung, and after smoking or environmental assault, we can see the holes in the lung not only is blackened, but also is start with the decline in lung function, which is FEV1, forced expiratory volume in 1 second. So, and that's the lung volume reduction surgery, where we have to remove certain part of the lung because some tumors in the lung. So I put an example here as the normal aging versus abnormal aging. Here is the age 40 to 50. We can see pretty healthy, and I'm in this range just now. And I would like to have a normal aging rather than the abnormal aging, where we have the oxygen mask, oxygen cylinder, to be able to give to the lung and open the lung so that they can breathe well. So this is effectively due to the tobacco smoking and also the biomass fuel burning, sulfur dioxide, other noxious gases. So we'd like to see how these changes occurs and whether we can stop the progression of these changes. So one thing, not only we can see the wrinkling of his skin, but and also the oxygen mask you see in these patients and taking inhaled steroids or bronchodilators, one of the important things we see as we age is the decline in lung function. And we can see if we climb these steps or go for hill walking, the smokers and people who live in those environment where they have more noxious gases, you see the rapid decline in lung function occurs, and the symptoms appears here when there is abnormal lung FEV1 measured as a forced expiratory volume in 1 second, and there is a normal decline which also occurs. So I focus on sirtuin because sirtuin is an important molecule which has been shown to be involved in anti-aging based on Drosophila, C. elegans, and mouse studies. So here we heard two aspects of sirtuin. One was the NAD and NADH ratio, and the other aspect I will briefly allude, which is a PARP1 activation, because genotoxic agents such as tobacco smoke and other gases can potentially break the DNA, and to be able to repair, which is a joining of the DNA with homologous, non-homologous joining and leading to cellular senescence, that's something we are studying and would like to mention in brief those aspects. So first one is sirtuin. We need the NAD to be able to function, and the protein, they are acetylated. Sirtuin deacetylated to protein release nicotinamide, and then we have the ribose as the byproduct. So there are several activators and inhibitors of SIRT1. One of the important activator which we found it out was resveratrol, and basically is a non-specific SIRT1 activator, and there has been a lot of controversy on that aspect too. So there are 7 sirtuins. Sirtuin 1, which is what we are interested, and basically it has the function of the deacetylating certain molecules such as NF-kappa B, P65, P53, FOXO3, and other substrate involved in survival, differentiation, and metabolism. There are other sirtuins. Certain sirtuins are mitochondrial based, and certain are in the nucleus, and some sirtuins are in the cytoplasm. So we have a colleague showing SIRT6 in DNA damaging response, and is hopefully that paper is coming out in Science in a couple of weeks. So these sirtuins are all important, but we target here SIRT1 to see how this is related to lung responses, particularly by tobacco smoke. So we hypothesize that the smoke has oxidative stress components and carbonyl content, which is the aldehydes and aromatic polyaromatic compounds, and then causes oxidative stress leading to lung inflammation and premature aging, which are associated with decline in lung function and lung airspace enlargement in the lung, which is the airspace in the lung become enlarged, and therefore they lose the lung function. And one aspect of this is the emphysema. The other aspect, which also is broadly categorized as chronic obstructive pulmonary disease, and certain patient with asthma also has the COPD. So asthmatics who are not responding to bronchodilators or steroids, potentially they can be a patient with COPD. And there are two hypotheses, British hypothesis versus Dutch hypothesis. British regards as two different diseases. Dutch people say is only one disease. Americans, they follow the British definition that there are two distinct diseases. SIRT1 regulates premature aging of the lung. So SIRT1, as we know, that is not inhibited by trichostatin A, whereas these class 1 and class 2 deacetylases are inhibited by trichostatin A. So something unique about these deacetylases. So SIRT1, as we see, that is deacetylate protein molecules as well as the histones, particularly histone lysine at 56 residue, histone 3, K56, in transcription factor NF-kappa B at 310 residues, as we see here. So this is a protein deacetylase, histone and the other protein. So it is regarded as the deacetylase, NAD-dependent deacetylase. So it has the nuclear localization signal because it stays in the nucleus. It can be transported out of the nucleus. It has an important residue at 366, and that's the target of the NAD binding, where it binds and imparts its activity. If you mutate this residue, you lose the ability of the sirtuin to be active. So we would like to see whether the resveratrol and ethanol, as we heard, can act on this particular molecule. And cofactor, we need NAD. Activators are resveratrol. Sirtuins make a compound which is based on resveratrol, SIRT1720, and that's merged with GSK, and that's another activator. It activates a nanomolar concentration, and inhibitors are nicotinamide and sirtinol. So SIRT1 basically is regulating inflammation, and this has a link with apoptosis, autophagy. It regulates FOXO3. We have a paper coming out in Journal of Immunology how this regulates and causes the lung inflammation, lung aging, and people have shown, other investigators have shown P53 regulation. And then we propose that SIRT1 actually regulates cellular senescence via P53 and P21, but there is another event which is a replicative senescence, which is a normal turnover of cellular division, whereas the premature senescence is something which is stress-induced. And previous speakers were highlighting the nun study. I would like to see whether if they are put under stress, what happens to them, whether they with their aging. So this is stress-induced premature senescence. There's something associated with abnormal aging. So we published several papers, and this is also corroborated by other people. I'm not going to go in detail about those studies. Basically, what we showed, tobacco smoking, oxidative stress alters sirtuin through post-translational modification. So SIRT1 is decreased in the lung cells as well as in the heart in response to tobacco smoking, and that is associated with inflammatory response, particularly NF-kappa B activation. And similarly, you can see another study in FASEB journal by investigator at Imperial College. They published in London. They published this is important and decrease in patients with bronchitis. So just brief, you can see the level of SIRT1 in smokers slightly diminished, whereas in patients with COPD it's dramatically decreased. And this is a staining of the lung airways and the alveolar region. We can see the reduction in sirtuin levels in patients with COPD. Less colors here means less SIRT1 levels. And this is another study in FASEB journal. They show with the progression of the disease, you see the reduction in SIRT1 level. And in mouse, when we hit with tobacco smoke, we put in a chamber, we can see reduction, complete disappearance of SIRT1 in their lungs. And similarly in rats, we see the reduction here. And in aorta, we see the reduction in SIRT1 levels. So SIRT1 is responding to oxidative stress somehow, and is perhaps redox regulated. So we pursued this phenomena, and this is another thing, we also treated C. elegans, smoke, though their membrane, cutaneous membrane is impermeable to the tobacco component, but still perhaps they were under stress, and they got the reduction in sirtuin. So SIRT1 regulates NF-kappa B, as I showed, is interacts with the deacetylase with REL-A 310 residue at lysine, and this is a cartoon showing that the SIRT1 is deacetylating P65, and hence it regulates inflammatory cytokines and chemokines. So this is an experiment we conducted where we showed the acetylation of REL-A in response to tobacco smoke extract, and this is associated with the acetylation and the similar link to the cytokine IL-8 and IL-1 levels. And when we have the siRNA of SIRT1, you can see the slight increase in the level of the acetylation of P65. When we treated the cells with cigarette smoke extract, we see further increase. So suggesting that SIRT1 regulates P65, and if you increase the oxidative stress, particularly by tobacco smoke, can lead to further increase in acetylation, and this was associated with cytokine release such as IL-8. So SIRT1 has some activators, and one of the co-substrate is cofactor is NAD, and there are inhibitors, nicotinamide, sirtinol, splitmycin, suramine. We use sirtinol because one of the effective inhibitor of SIRT1, whereas we have these non-specific inhibitors too, splitmycin and suramine. So we carried out one experiment to see whether activators and inhibitors can alter the cytokine levels in mononuclear cells, and you can see the smoke increased the cytokine here, and resveratrol was able to inhibit to certain extent, and high concentration 5 micromolar, you can see further reduction. Sirtinol actually further augmented the response, and similar thing happens for cytokines such as TNF. And when we use this compound, we synthesized commercially, and we were able to see much more dramatic reduction of cytokine levels using nanomolar concentration of SIRT 1720. And similar thing happens in previous publication that resveratrol can also reduce the cytokine level from macrophages. So SIRT1 undergo post-translational modification. One thing is by kinase signaling, and the other part would be the carbonyl modification. And other aspect will be the depletion of NAD, which we heard that NAD is also important in regulation of SIRT1. So we assessed, because tobacco smoke contains carbonyls, acrolein, and acetaldehyde, and whether that can directly bind with SIRT1 and can lead to post-translational modification and proteasomal degradation. So we found that the 4-HNE, 4-hydroxynonenal, and also 3-nitrotyrosine, when we did the immunoprecipitation of SIRT1, we were able to show that these molecules interacted with SIRT1 and also increase in patient with COPD, which is their lungs, suggesting that this SIRT1 undergoes post-oxidative, post-translational modification. I wouldn't use the word oxidative, it's particular because I don't have a direct evidence, but it's mainly the carbonyl modification here. So similarly, we can see SIRT1 in cells treated with these compounds. The tobacco smoke can cause the post-translational modification. At the same time, it can cause the phosphorylation, and the phosphorylation event is reversible, but when phosphorylation and carbonylation occurs, these proteins, they are damaged so much that it undergoes proteolytic cleavage. And one of the assays we use is the DNP, and this is just the cartoon showing how the assay works. And we were able to show that the DNP was increased in response to tobacco smoke, suggesting that again there is a thiol group modification going on. So we looked the SIRT1, whether this is a modified on cysteine residue by carbonylation. We did mass spec and all the proteomics, and we were able to show a group which was modified. Perhaps this is just showing the proteasomal. When we use the ALNN, which inhibits the proteasome, we were able to rescue the SIRT1 degradation here, but the activity was decreased, though we recovered the SIRT1. So just to allude that the important substrate, which is NAD, has bifunctional events here. It requires not only for SIRT1 activation, it also is associated with PARP1, and NAD is a substrate for PARP1. When the PARP1 is activated, it utilizes NAD, and PARP1 is a response to DNA damage. So we looked these phenomena in response to tobacco smoke, and we were able to show that the PARP1 is activated by tobacco smoke here in response to the cigarette smoke extract and respect to the hydrogen peroxide, and these PARP1 inhibitors actually able to inhibit the response. So just to show quickly in the cartoon that SIRT1 requires NAD. When NAD is depleted, PARP1 is activated, SIRT1 undergoes carbonylation reaction. So polyphenol, perhaps I will skip this because due to lack of time, and I would go back to this mouse study, just a couple of more slides, whether I have a couple of minutes, only 1 minute, as the chairman says. This mouse study, basically what we showed is the SIRT1 deficient mice, they age faster and they have the lung emphysema, whereas SIRT1 overexpression protects against the tobacco smoke response. So, and same thing happens to exercise tolerance, that these mice are not able to run faster as we expected. We would like to see whether we can put the resveratrol in the lung, whether we can fill with the airways with the oxygen. And I'm not going to go about DNA damage response. Basically, this is to conclude the presentation that we have seen SIRT1 is reduced in response to tobacco smoke oxidative stress in several models, and this is associated with FOXO3 acetylation, NF-kappa B activation, and this is involved in several processes, including the premature aging of the lung. And with this, I conclude. And postdoctoral fellows, graduate students, some collaborators, and one of the collaborators, we have Dr. Frank Antolcioni. He's from University of Reims, and this is funded by the NIH federal grants and through R01 mechanisms. So thank you so much for listening.
Thank you very much, Dr. Rahman, for this detailed presentation. Now we have time for a few questions, please.
Thank you, Doctor. I have a question. You showed in one of your cartoons that SIRT1 is oxidized in the nucleus and then coming into the cytosol and degraded. Is there any evidence that it's not degraded by the proteasome in the nucleus? This transport mechanism is interesting for
Yeah, I skipped those slides actually. There is a nuclear localization signal, CRM and RN, and we don't know what happens, but actually SIRT1 shuttles and tobacco smoke reduces the shuttling. So the proteasomal lysis degradation can occur in the nucleus. It's possible. And we do see reduction in the nucleus. So, but we have not looked. Well, we have one experiment. We did leptamycin, which inhibits the shuttling, and we are able to show the degradation. So that means it is degraded in the nucleus and through the proteasome. So one has to perhaps know whether proteasomes are present in the nucleus. So something you would like to address?
Oh, yeah.
Yes, they are present. So,
Okay.
Irfan, I have difficulties establishing an activity for SIT1, and in quite a few of the slides you refer to expression, and actually that's not activity. What we are doing, because we are in destination, is looking at the acetylation levels of TGC1 alpha, and that might reflect somehow SIT1 activity. But concerning your experience in subchronic exposure, 8 weeks, yes, we do not see any changes in NAD or NADH. But what we see are changes in NADPH that I think you published. Yeah. And that will favor this pentophosphat pathway and also the synthesis of the antioxidant enzymes. And there is a discrepancy here in an oxidative stress that might occur, yeah, and which is necessary for the activation of the circuits, and at the same time the activation of the dairy elements and the antioxidant enzymes. Can you comment on this?
yeah, so the first part is whether SIRT1 activity is reduced in response to subchronic and chronic exposure to tobacco smoke and other pollutants. Yes, actually the activity is reduced, and potentially that can be due to several factors. One of the factors which I mentioned, due to post-translational modification and subsequent degradation. So that's one part. Second part, the question was whether the defense mechanism can be augmented. For example, antioxidant mechanisms, catalase, SODs, and glutathiones, GCL. Initially, smokers, they do have adaptive response to counteract the radicals, as you know. But I think at the later stages, particularly the studies published in field of NRF2 in smokers and patients with COPD, the level declines rapidly, and hence the counteractive mechanism is not there, and hence these overwhelm these pathways in these subjects.
8-week exposure cannot be equated to COPD.
Yeah, so I didn't go too much into the 6-month study, and due to lack of time and short of time. So 6-month studies we have done where we see the emphysema in mouse, and we see similar thing, and that's hopefully will come soon. You will see in public literature. Yeah.
Any more short question? Yes, the short one, please.
Do you think the ratio between NAD and ADH in the nucleus is the same as in the cytosol, or you have some? How is it regulated? Because
no, I don't have that idea whether, but NAD is.
Do you think NAD and ADH pass through in the nucleus, or you have something?
I have not done that kind of experiment, so maybe the previous speaker can address.
The ratio of the nucleus is the same as cytosol. The ratio in mitochondria is 3 orders of magnitude different. The ratio nuclear is